Hello girls, good morning everybody. Welcome back to these video classes. So since this is our first class, I'll be telling you about the syllabus. I'll just brief you about the syllabus in this video. Okay, so now our topic is genetic engineering and tissue culture. So now, what do you mean by genetic engineering? Genetic engineering means it is the manipulation of the genetic material. What is the genetic material? The DNA or RNA of an organism. This is the genetic material. Whatever genetic material the organism has. So we all eukaryotes have DNA as genetic material, even the prokaryotes. Viruses have RNA as their genetic material. So engineering with the genes is called as genetic engineering. And tissue culture, culturing of tissues, culturing of tissues. Here in botany, we will be culturing the plant tissues on artificial nutrient medium. That is tissue culture or plant tissue culture. So I'll just brief you about all the topics in our syllabus in this SEM. So the first topic is concise account of methods used in DNA technology. The DNA technology. What do you mean by DNA technology? It is nothing but recombinant DNA technology or rDNA technology. That is what I told you till now. That is the manipulation of genes. So concise account of methods. What are all the methods used in the recombinant DNA technology? And then restriction enzymes. These are some scissors. We call them as molecular scissors used to cut the DNA at particular sites. So restriction enzymes and ligases. These are molecular glues or gums to join the DNA fragments. Cloning vectors. Vectors are carriers. Say for example your bus which carries you from one place to another place. That is a vector. So like that there are cloning vectors used in rDNA technology. So we will be studying about those. So methods on rDNA technology, restriction enzymes or molecular scissors, and ligases or molecular glues, and cloning vectors. Then we will come to the construction of rDNA, construction of recombinant DNA or hybrid DNA, because there are two pieces of DNA in this gene, in this genome. So in the genome of a particular organism, we have introduced a foreign gene. Now that organism is a recombinant organism or a transgenic organism because it has genes from another organism. So it is called as transgenic organism. So construction of this rDNA with taking examples of NIF gene, nitrogen fixing gene present in plants, and the cDNA libraries. We will be studying about the complementary DNA libraries. And then we have a topic called brief account on hazards and safeguards of rDNA technology. Now this recombinant DNA technology is quite a new branch in biology, in science. So we don't know in the long run what are its hazards and what safeguards we have to do about it. So all this comes under the topic hazards and safeguards of genetic engineering or rDNA technology. This is about the genetic engineering topics. Next, coming to tissue culture. So in tissue culture, I told you a plant grows in its natural habitat outside in the soil. So then that is in the natural habitat in vivo. Same plant can be grown in the lab, which we call as in vitro. So in vitro, if we have to grow a plant, what are all the techniques? What all do we need? All this comes under the branch of tissue culture or plant tissue culture. So we have techniques of tissue culture, and then differentiation, how from that small bit of tissue how a plant differentiates. We can bring any small piece of the plant which we call as explant. The small bit what we bring to the lab to grow culture is called as explant. It may be a root bit, piece of a root, or a piece of a stem, or leaf, or flower piece, or fruit, or anything, any part of the plant which we call as explant. We bring it to the lab, clean it, sterilize it, and then we prepare an artificial nutrient medium. Medium means what? The support system, the substrate for the growth of the plants in culture. So what all the plant gets in vivo or in the soil conditions, what are all the nutrients the plant is getting, what all nutrients the plant gets, same nutrients are also added and a medium is prepared for the growth of the plant. So the plant is grown in a tissue culture lab. under controlled conditions on the nutrient medium. So we learn about differentiation. There it differentiates. The explant differentiates and forms a callus, and then it forms a bud, shoot bud, like that it grows. So the differentiation we learn. Totipotency. Totipotency means what? A plant cell, a small plant cell is capable of regenerating a whole new plant. This ability or the potential of the plant cell to grow into a whole new tree or whole new plant is called as totipotency. This is seen only in plants. Animals don't have totipotency. So we study the concept of totipotency. And then organogenesis, how all the different organs are initiated in the culture. And then we have protoplast isolation and culture. What do you mean by protoplast? Protoplast is the in leaves. There are cells in leaves, and when we remove the outer cell wall, what remains inside is called as the protoplast. Protoplast means the inner contents. Cell devoid of cell wall is called as protoplast. What do we see there? The cell membrane and all the other inner cell contents. So all together we call it as protoplast. So we can isolate the plant protoplasts, mostly the leaf protoplasts, and then we culture it, try to grow it. Okay, so protoplast culture. And then somatic hybridization means what? We are taking protoplasts of two plants and then we are trying to fuse them in vitro in lab conditions. We are trying to fuse the protoplasts. So the fusion of protoplasts is called as somatic hybridization, and from that somatic hybrid we can produce a new plant. So like this, in vivo, which plants are not able to cross, and breed, those plants we can cross them and breed them in the lab conditions. We can cross. So distant crosses can be made through somatic hybridization or protoplast fusion. So we will be studying about that. And then the synthetic seeds. You know what a seed is. So such seeds are prepared artificially in lab conditions. So those are called as synthetic seeds. So there will be embryos, somatic embryos, which are encapsulated in a gelling medium, gel-like medium, which has all the nutrients and protectants for the embryo to survive. And these are called as synthetic seeds. So this we can produce in large number, in mass, and we can handle it also very easily like any other seed, and it can be grown. So that is synthetic seeds. Synthetic seeds. And the last topic is haploid production. Haploids are plants which have single set of genes, single set of chromosomes. So such haploids can be produced through tissue culture. Haploid plants means they are true to the mother. Whatever genome the mother has, same, because only from a haploid set of chromosome it is produced. So same genes. So they are nothing but clones of the parent plant. So haploid production also we will study with reference to androgenesis and gynogenesis. Androgenesis means from the male parent. So from the pollen grains, anther or pollen grains, how a plant can be cultured or cultivated. Gynogenesis is from the female parent, the female parts. From that, how a whole new plant can be regenerated. So we will be studying about haploid production, and in that androgenesis and gynogenesis and its significance. So this is our syllabus for this sem. So what is our syllabus? Genetic engineering and tissue culture, plant tissue culture. So then we will meet in our next class.